Okay, so for this lab, we're going to look at fuel utilization during exercise. And here we want to answer, how do we know what substrate we're using to form ATP? We've kind of gone with this assumption that we're using glucose. Glucose is really the fuel that's key here. But we also know we use fats. We also know we potentially could use proteins. So how do we know? And is that the same during all types of activity and during lengths of activity? And so the answer is yes, is that we can at least indirectly figure out what this is. And so a term we're going to use quite a bit, in fact, in lab, we'll look at this actual in real time, is this idea of what's called respiratory exchange ratio. So let me just give a brief definition of this. This is simply going to be the volume of CO2 divided by the volume of oxygen, VCO2 over VO2. Right? Now, the CO2 being carbon dioxide, O2 being oxygen, and it's going to be the volume of CO2 that we're expiring divided by the volume of VO2 that we are inspiring. Now, why is this important? Well, this will give us some value. In fact, there's a very limited range of values that it can actually give us. What we know is that if the RER value is 0.7, in other words, VCO2 divided by VO2 gives us the number 0.7, we know that we're using exclusively fatty acids to form ATP that we're utilizing during exercise. On the other hand, let's go to the other side of this. If our RER is 1.0, in other words, VCO2 and VO2 equal, then we know that we are exclusively using carbohydrates to form that ATP. Now, in reality, it is very, very rarely one of those. Usually we're looking at a number in the middle, somewhere between 0.7 and 1.0. And in lab, we'll see this. We'll see that we can actually measure this very easily. We put a mask with a rebreathing valve on, we strap it around your head, we connect that to a tube, so all the air you expire, we can collect and we can analyze that. We can also figure out the volume of air you're inspiring, and from those we can make some calculations. We have an O2 and CO2 analyzer. We can make some calculations and figure out what VO2 and VCO2 actually are. Now, these are indirect measures. I mean, really, if we really wanted to know, we would go into each cell and see what substrates are being used, or maybe we could go in the blood and measure the CO2 content in the venous blood and the O2 content in the arterial blood. Well, obviously, that more direct measure is not easy to do. It can be done. We can take blood samples if we wanted, but this way, what that we call indirect calorimetry, indirect measurement, is still useful, and it is directly correlated to what's going on inside the cell and ultimately what we can measure inside the blood. Now you may be asking, so where do these numbers 0.7 and 1 come from? They seem pretty arbitrary. Well, it actually goes back to your friend and mine, the biochemistry. If you recall from nutrition, or if you recall from when we talked about glycolysis, glucose has a structure of C6H12O6, each of our carbohydrates. Now, asterisk, I know some of you are like, well, what about sweeteners? Some of them have different compound structure. And you're right. Let's ignore that for now. So all carbohydrates, C6H12O6. Fats, here we're talking about triglycerides, are C6H32O2. So these are different chemical structures. Again, all have carbon, hydrogen, oxygen, but definitely different chemical structures. Well, why is this important? If we utilize these and break these down, right, if you recall what we talked about with glycolysis, what we talked about with the Krebs cycle, if we follow the path of a glucose molecule through those steps to forming ATP, if we follow a fatty acid through those steps that fatty acids use in beta oxidation, if we follow those steps to what forms ATP, the byproducts of those for carbohydrate are 6 CO2 and 6 water. Okay. However, for fats, the proportion is slightly different. If we're going to oxidize a fat, we actually need 23 O2, and that gives us 16 O2 and 16 water. So if you begin to do the math here, if you look at these ratios, well, for carbohydrate, 6 oxygen, 6 carbon dioxide, 6 divided by 6 is 1. If you look at our fats, 23 oxygen, 16 CO2, do the math, it's 0.7. And so that's where we get these numbers from. And it's not just arbitrary. These actually come specifically because of the chemical structure and when we're oxidizing these macronutrients to utilize them to form ATP. All right, so what's the benefit of knowing this? Well, here's the benefit. We know that during different types of activities and different intensities and durations of activities, we actually prefer one type of substrate over another. So generally speaking, and I just be very general on this, during low intensity exercise, now we're going to say low intensity exercise, less than 30% of VO2 max, we generally primarily like to use fatty acids to form that ATP. So not surprising, if we are engaging in a low intensity exercise like walking, something that's not terribly intense, we wouldn't be surprised to see that the RER is probably closer to 0.7. Now, it's probably not going to be exactly 0.7. It'll be 0.78 or something like that. On the other hand, we know that during higher intensity exercise, right, where we require more ATP, where potentially we may be producing lactate, potentially, we know that in this type of activity, we actually primarily use carbohydrates. And because of that, the RER starts to shift to 1.0. Now, a couple of things to understand here. When we look at this number of 0.7 or whatever, 1.0 or some number in between, we're looking at the entire body. Again, we're looking at air that's being expired. Well, that respiration is a consequence of what's going on in every cell in the body. And that's important because just because we are measuring an RER of 0.82, right, doesn't mean every cell is utilizing that certain percentage of fat and that certain percentage of carbohydrates. Doesn't mean that at all. It could quite well mean that some cells, if you could measure their individual RER, would be more like 0.7. Others might be closer to 1. So this RER, when we measure it indirectly, is really almost a summation of all the cellular respiration that's going on. And with this, we have this term, and your book uses this term quite a bit, called the crossover effect. This idea that as we change in intensity of exercise, we begin to cross over from using more fats to more carbohydrates. And if you remember back to forming ATP and the benefits, the pros and cons of glycolysis, the pros and cons of the Krebs cycle, this kind of makes sense, right? With low intensity exercise where we don't require as much ATP so we can form it more slowly, fats is a pretty good choice. As we start to increase in intensity where we now need more ATP, doing it anaerobically where maybe not as much oxygen is available, ramping up glycolysis, therefore doing this using more carbohydrate, is a little better choice. Now, the reality is, even in high intensity exercise, we're still using fats, right? It's not like there's this valve that we go from one to another. It's a gradual crossover from one substrate to another. So the question is, why do we care? I mean, all right, it's nice to know if I'm walking, if I'm jogging, if I'm playing whatever sport it is. Yes, sure, it's nice to know that, I'm using 30% fats and 70% carbohydrates. Well, you can probably imagine that for some individuals, let's say of someone who wants to reduce their percent body fat content, right? Admirable goal. You could argue that they would want to engage in an activity that would utilize more fats. Well, then you'd probably say, well, low intensity activity. In fact, it's a fairly common recommendation. If you want to lose weight, lose body fat, we often say long duration, low intensity exercise. And that's where this comes from. It comes from the metabolism and what the substrates are to form ATP. Now, you could take that to an extreme though. I mean, you could say, well, if you really just want to use fats, you should have like really, really low intensity exercise. In fact, maybe don't exercise at all. And then you would be using exclusively fats. And I hope you get the thinking there isn't exactly productive. I mean, the sedentary lifestyle is not the best choice. So there's an extreme, obviously, to the utilizing fats. And the reality is we do during long-term exercise use fats just by the nature of the activity. And as we talked about in class with oxygen debt epoch, we also use those fats in those times of epoch, which occur after exercise for all individuals. And so I want to, don't buy too much into the, if you want to burn fat, low intensity exercise, that's the way to go. That's not wrong. And in fact, it reduces injury. There's value to that, and particularly in a more older, elderly population, but it's not necessarily the best means for at least weight production. Okay. Another thing to understand in this is that if you recall way back in glycolysis, right, we needed something to start this process. Well, what was that something? That something was ATP. So we need ATP, remember, to initiate glycolysis, to initiate at least utilization of carbohydrates, and honestly, even also to initiate beta oxidation of fats. So we often say that, okay, even in burning fats, you still need carbohydrate to initiate this process. And so your book uses the term fats burn in the flame of carbohydrates. It's kind of a little kind of corny term, I guess. But yeah, the idea that you do need carbohydrate present to utilize fats ultimately to form ATP. And this is key because with longer term exercise, and when I say longer term, we're talking an hour, more than an hour, where we might start to deplete muscle and liver glycogen, especially muscle glycogen, that carbohydrate may not be as readily available due to reduced rates of glycolysis, issues with some of the enzymes in both Krebs cycle and glycolysis, inability then to utilize fats. So this may seem like not such a big factor, but during long-term activity where we may come to a point where fats are our most available source to form ATP because we've depleted carbohydrate, doesn't mean we still don't need carbohydrate. Doesn't mean we don't still need to take that in. And this is one of the reasons why during activity we take in carbohydrate an hour in. We want to take in some form of glucose or something. Now with this, and what I want to end with, we've obviously ignored protein. Okay? And we can form ATP from amino acids. We saw that back in metabolism. There are pathways for amino acids to be broken down, gluconeogenesis, to then form ATP. But that is not the preferred pathway. In fact, truth is, very little of the ATP you form in normal circumstances. Notice I said normal circumstances. There are situations where we may actually really break down proteins to do this. Extreme fasting, some other metabolic disorders, we might see that. But otherwise, protein plays a very small contribution to this ATP formation. And that makes sense. I mean, we need these amino acids, these proteins, for numerous other functions. Muscle contraction, they play a role obviously in cell membranes, going to play a role in bone, other key structures of the body. So they're not necessarily a great substrate to form ATP for extended periods of time. Now, having said that, we still do it. In fact, some would say, and I think there's truth to this, it's not surprising that if you look at runners, you look at individuals who engage in really long-term exercise where they're really depleting carbohydrate stores, not surprisingly, they generally are not the most muscular individuals around. Now, is that because they're using protein to form ATP? Maybe. Is that because they're engaging in activities that don't require as much muscle mass? That's probably the case too. But there may be some truth to that of sources of ATP that we use. So lastly here, I just want to look at this graph looking at exercise duration and what sources we're using. And as you can see, as we're depleting this muscle glycogen, and now this is over time, that there are other stores, free fatty acids, triglycerides, fats, lipids here, blood glucose if it's available. There's a big key to that. If it's not, this really is non-existent. But as we're increasing in duration, we are starting to use more and more fatty acids, irregardless of the intensity of the activity. And I think that's key. If carbohydrates are not available, even if we're engaging in a high intensity activity, our only source left is fats. Not surprisingly, it would make engaging in that high intensity activity fairly difficult. Good place to pause. We'll see you in class in the next lecture.